Now next we'll move towards RNA interference. So this interference is the mechanism of gene silencing. Okay, you can switch off expression of particular gene using this technology, RNA interference. This is also known as post-transcriptional gene silencing, because here after transcription the mRNA molecule is silenced. Silenced in the sense its expression is turned off. Okay, gene expression can be silenced by some small RNA molecules. This phenomenon has been termed as RNA interference. It was first seen in plants but later found in mammals, including human beings. RNA interference is an RNA dependent gene silencing. This RNA causing gene silencing may be endogenous RNA or exogenous. Endogenous, that is cell which has its own RNA, it is used for silencing, or the RNA which comes from the different sources like from viral infection into the cell, that RNAs can be used for gene silencing. Those RNAs are known as exogenous RNAs. Endogenous RNA that can silence gene is known as microRNA, that is miRNA. Exogenous RNA that can silence gene are known as siRNA, that is small interfering RNAs. These RNAs are short, of near about 21 to 20 nucleotide long. siRNA can be of viral origin or it may be synthetic, used as drug for silencing expression of particular gene. miRNA discovered by Ambrose et al, that is the scientist which helped this Ambrose, which worked with this Ambrose, that means et al, in a nematode C. elegans, Caenorhabditis elegans, which is a nematode used as model system. This organism is fully transparent. You can actually view various pathways that are occurring inside the body of this Caenorhabditis elegans. Fire and Mello discovered double-stranded RNA causing RNA interference in Caenorhabditis elegans. The double-stranded RNA came to be known later as siRNAs. Both si and miRNAs are formed from large precursors, larger RNA molecules. They are cut to form short siRNAs or miRNAs, depending on their origin, whether they are exogenous or endogenous. Now the siRNA. The precursor of siRNA is the stem loop structure like this, which is shown here. This is cleaved to form double-stranded small interfering RNAs. Cleavage is catalyzed by enzyme dicer, which has ribonuclease activity. Means the enzyme which can cut RNA. This dicer belongs to ribonuclease 3 family. Remember, siRNA is cleaved by dicer. siRNA is 21 to 23 nucleotides long. 3' end of each strand has overhang and the 5' end of the other strand has overhang. Means each strand has overhang of 2 nucleotides. Okay, one strand's 3' end has overhang and other strand's 5' end has overhang. That means this is like a sticky end generated by restriction endonuclease. Okay, this is not generated by restriction endonucleases. I am giving you an example of that. Remember, siRNAs has overhangs and size of siRNA is 21 to 23 nucleotides long. siRNA is integrated into a protein complex in an ATP dependent reaction. The complex is termed as RISC, RNA induced silencing complex. Okay, siRNA, or say this is a RISC complex, binds to siRNA. The active component of RISC, RNA induced silencing complex, are some endonucleases called argonaute proteins. Out of two strands of this siRNAs, only one direct silencing. This strand is known as guide strand. The other strand is known as passenger or anti-guide strand. Remember, only one strand out of these two. Now both SI and miRNAs are double-stranded molecules. Out of these two strands, only one is used for gene silencing. That strand is known as guide strand, and the other is known as anti-guide or a passenger strand. The passenger strand is degraded by argonaute protein, which is a part of RISC, RNA induced silencing complex. This results in activation of RISC, RNA induced silencing complex. The guide strand binds and binds to a complementary mRNA. Okay. Now when it binds to the mRNA, argonaute protein cleaves that mRNA. So this here, this is stem loop double RNA. The dicer enzyme, which has ribonuclease activity, binds to the stem loop precursor RNA and cut this RNA into short fragments of a size 21 to 23 nucleotides with 5' and 3' overhangs on each strand. Okay, this forms siRNA, small interfering RNA. Now to this small interfering RNA, RISC complex binds, which consists various proteins, and one important protein is argonaute. This argonaute digests other strand of siRNA. Now only one strand remains, and this one strand finds complementary sequence onto the mRNA. Okay, only to the mRNA this one strand or guide strand of mRNA siRNA binds, and then this argonaute cleaves mRNA. Now once the mRNA is cleaved, mRNA is not available for translation, and hence the genes present on this mRNA are silenced. Okay, and this is how this RNA interferes with the expression of the genes. So the technology is known as RNA interference. Synthetic siRNAs can be used to silence desired genes. This is used for various therapies, like if the person have particular disease and a protein is overexpressed in that particular disease, so such mRNAs encoding that diseased protein are targeted with these siRNAs. The siRNA should have a sequence complementary to the mRNA of the target gene. siRNA as drugs. siRNAs have been tried as drugs to silence specific genes in human beings. Some success has been reported in age-related muscular degeneration and cancer therapies. Now the problems with siRNAs as drugs. Several problems are yet to be solved in therapeutic use of siRNAs. Immune system can destroy this siRNA when it is injected into the cells, mistaking it as viral RNA. That is, when the siRNA is transferred to the human cells, which can target mRNA responsible for diseased protein, these siRNAs are recognized as viral RNAs and may be cleaved by the cell's defense mechanism. Homology in the base sequence of the genes can result in silencing of unintended genes. Maybe sometimes the genes which are not a part of disease, they may be silenced and can develop some other disease. Response of different types of cells to siRNA